Pretty much everyone knows that Frodo Baggins is a hobbit, but what you might not know is that there are three types of hobbits. Harfoots, like Frodo, Fallowhides, like Merry and Pippin, and Stores, including Smeagol, who, as we know, goes on to become Gollum.
Precious.
It seems clear to me that the different species in Lord of the Rings, and I'll assume that they are species, share some common relationships, so that we can assume that hobbits are closely related to humans, and perhaps more distantly related to the elves. There are also some things, like this Goblin King, that seem to be quite distantly related to the other creatures that we've mentioned. The various creatures in the Lord of the Rings share some kind of evolutionary history, so that we can make a tree like this, linking the different types of hobbits as close relatives and more distantly related to things like the Goblin King. What? That's the wrong Goblin King? Oh, yeah, sorry. We would then expect close relatives to share certain traits. For example, all of the hobbits have excessively hairy feet. But why would we expect close relatives to be more similar to one another than they are to distant relatives? And just how similar should they be? The way that we can think about that is by applying our mathematical models of evolution, like Brownian motion, onto phylogenetic trees. And the core question that we start out by asking is, why are close relatives similar to one another? Consider this very simple phylogenetics tree containing only two species. We can plot the phenotypes of these two species in a simple plot like this, with the phenotype of species 1 on the x-axis and species 2 on the y-axis. We start out at the root of the tree and think about evolution on this branch, which represents a common ancestor of species 1 and species 2. Since we have a single lineage that's ancestral to both species 1 and species 2, we can plot any phenotype that that species has along this dotted line, which shows the one-to-one relationship between the phenotype of species 1 and species 2. These two species haven't diverged from one another, and so their phenotype at this point is equal, and we can plot that with this blue dot. As the species trait evolves along this red branch, the value for species 1 and species 2 remains equal. Under our model, though, we assume that following speciation events, the two species will now evolve independently of one another. Now the value of species 1 can change independently of the value of species 2, drawing our blue line off of this red dotted line. What we expect then is that the blue dot will be off of the red line because of the independent evolution of species 1 and species 2. But especially if the two species have diverged from a common ancestor recently, that blue dot might not be very far from our red dotted line. The point that I'm trying to make here is just that species covary because they descend from a common ancestor. We can understand exactly how this covariation behaves under a Brownian motion model. When characters evolve under a Brownian motion on a tree, then the species ending values will follow a multivariate normal distribution, and variances and covariance of the species will describe exactly how they relate to one another. For example, if we consider the experiment that we just showed with our two-species tree, if we were to rerun that experiment many times and plot a cloud of points, it would look like this. Although the individual values of species 1 and species 2 differ a lot among replicate evolutionary trials, there's overall a correlation between the value of species 1 and species 2. In other words, if species 1 has a large phenotype, around 4, species 2 also tends to have a large phenotype. If species 1 is small, species 2 also tends to be small. This is because they share evolutionary history through their common ancestor. Even though they have had some time to diverge independently, the imprint of that shared evolutionary history remains in the data. Going back to our tree, under a Brownian motion model, the covariance of species 1 and species 2 we can calculate as the product of the rate of evolution, that's our sigma squared parameter, times the shared branch length of those two species. If we label that branch as a certain length, t1, then the covariance of species 1 and species 2 is just sigma squared times t1. One interesting thing about Brownian motion is that species 1 and species 2 will share that covariance of t1 no matter how long they have to independently evolve. In other words, if we were to extend these branches out even longer, they would still have a covariance of t1 times sigma squared.
We can also think about the variance of those species. So what we're thinking about here is if we reran evolution lots and lots of times and looked at the value of species 1 over each of those replicates, how much variance would we have among those replicates? So those replicates would all follow a normal distribution, and we're just describing the variance of that normal distribution.
The variance of species 1 is the rate of evolution times the total branch length from the root of the tree to that tip. In this case, T1 is the length of the common ancestor, T2 is that branch leading to species 1, and so the variance of species 1 is sigma squared times T1 plus T2. Similarly, the variance of species 2 is sigma squared times T1 plus T3. What we have here then is a covariance between the two species and a variance for each species. We can put these terms together into a matrix called a variance-covariance matrix. This matrix shows the variance of each species along the diagonal and then the covariance in the off-diagonals. For example, species 1 has a variance of sigma squared times T1 plus T2, and the two species have a covariance of sigma squared times T1. One way to understand this process a little bit better is to think about simulating evolution under a Brownian motion model on our tree. Let's imagine that we start out with a root character value of 0. The way to simulate Brownian motion evolution on a tree is just to simulate the changes that occur on each branch. For example, along our root branch, perhaps we have an increase in our character of 1.2, and then along our independent evolutionary branches, we have a change of negative 0.4 in the branch leading to species 1 and 2.1 in the branch leading to species 2. We can then traverse the tree, adding those changes together to figure out our phenotypes for species 1 and species 2. We have 1.2 minus 0.4 equals 0.8 as a value for species 1, and likewise 1.2 plus 2.1 is 3.3 for species 2. This is one way to simulate evolution on a tree, but it's really actually kind of slow and cumbersome. Equivalent to that simulation, we can also just draw a number from a multivariate normal distribution following this variance-covariance matrix, and under a Brownian motion rate of 1, we would have a variance of 3 for each of the two species and a covariance of 1.2. We also need to know our means, and our mean is just our starting value here. This goes along with the idea that Brownian motion has no expected trend, that the expected value at the end of the process is equal to our starting value. And so we can simply draw two numbers from our multivariate normal distribution. For larger trees, the pattern is exactly the same. Our tip data will follow a multivariate normal distribution that we can describe with a vector of means, all of which are equal to the starting value, and a variance-covariance matrix taken from the shape of the tree. The only thing that happens with bigger trees is that our matrices get bigger. For example, in a phylogenetic tree with 4 species and these branch lengths plotted here, we have a 4x4 variance-covariance matrix, and the elements of those matrices are the same as what we had before. So if we look at two species, x1 and x2, that's our first 2 rows and columns of the matrix. We can find their covariance, which is the branch that they share. So x1 and x2 have that common ancestral branch, which is red, has a length of 1.0, and their covariance is 1.0. x1 and x3, if we've traced their common ancestry, it goes all the way back to the root of the tree. They have no shared branches, and their expected covariance is 0. So we have this variance-covariance matrix that completely describes the distribution of tip states. If we wanted to simulate evolution on this tree, again, we could simulate changes along each branch and add them up to get the species values, or we could just draw a number from this multivariate normal distribution with the variance-covariance matrix shown here. Let's use this multivariate normal distribution to understand how we can calculate the likelihood of a continuous character evolving on a phylogenetic tree under Brownian motion. Again, we're going to think about variance-covariance matrices, and we'll call this matrix that we derive from the branch lengths of the tree and its topology C. So what we're going to assume is that we have a variance-covariance matrix C for some phylogenetic tree, a Brownian motion model with parameters z bar 0, that's our starting species mean trait. The bar over the z is just denotes that we're thinking about species mean traits rather than the traits of individuals, and sigma squared, that's our Brownian motion rate parameter. We also assume that we have species means for all taxa in the tree, and that's going to be denoted with this x. And x in this case represents an n-dimensional vector, where n is the number of species, and we have a mean for each species in the tree. Given all of those things, we can write down an equation for the likelihood of our data. So the likelihood of our data x, given our model, which is the 2 parameters of the Brownian motion model, and c, which describes our phylogenetic tree, is given there. And that's a complicated equation from matrix algebra. It looks complicated, but actually all that equation is saying is that the likelihood of x, given our model, is the probability of drawing a vector x from a multivariate normal distribution that's described by c
the variance-covariance matrix, multiplied by sigma squared, the rate parameter, and a mean vector described by z bar 0. So what's the relationship between our matrix C and our parameter sigma squared? Well, basically, sigma squared is a multiplier that stretches or shrinks the branches in our phylogenetic tree, and that stretching and shrinking then gets translated down into our variance-covariance matrix. We can visualize that here. We have a number bar for sigma squared at the bottom and our phylogeny there at the top. If we increase or decrease sigma squared, it effectively makes all the branches in our phylogenetic tree bigger or smaller. We can think about that in terms of a plot for species 1 and species 2, which again covary because of their shared branch length. And if we increase or decrease our sigma squared parameter, we basically rescale that evolutionary change, changing the covariances and variances of the species. Okay, so how does this work in practice? Well, here's a data set that's famous in comparative biology because it was used in a paper by Garland et al. In 1992, which is really one of the first quantitative comparative methods papers ever published in systematic biology. The first column that we have there shows the body mass of the species in kilograms for a whole bunch of different mammals. We also have a column showing home range. We'll talk about that later. So what we're going to think about is fitting a model of Brownian motion evolution to body mass. But we're going to use log transform body mass for reasons that I'll explain in a different video. We can use our likelihood formula to calculate the likelihood of that data given a model. And our model has two different parameters, sigma squared, which is the rate of evolution, and z bar 0, that's our ancestral character state for log body size for our mammals. Now, here we have the whole likelihood surface plotted, but when we're trying to maximize the likelihood, we don't know what that surface looks like. Instead, we can just try a value. So here I have a blue dot representing the likelihood of our model where we have ancestral to state of 0, so our ancestral body mass of 1 kilogram, and a sigma squared value of 0.1. The computer algorithm that maximizes the likelihood then tries a few points around that blue dot, decides which direction is uphill on our likelihood surface, and moves to a new point and another point, hopefully getting closer and closer to the optimum each time, until we arrive at the peak in our likelihood surface right here. And in this case, that peak gives us our maximum likelihood model parameter estimates, z bar 0 of 4.64 and sigma squared of 0.088. So now we can answer the question of how similar we would expect the two types of hobbits to be. It depends on the length of the branch of the phylogenetic tree that they share. At least under a Brownian motion model, that length, along with the rate of evolution, determines their covariance. So hopefully that gives you a little bit of insight into how these models behave when we think about them on trees, how we can use them to analyze data. Next time I'll talk to you a little bit about how independent contrasts work and the relationship between independent contrast and maximum likelihood fitting of models on trees.